All right, so let's carry on with our discussion about viruses. This is where we ended off. We talked a little bit about viruses and their size, the fact that they are not actually considered to be living entities. And this is where we finished off. We were discussing the host range of viruses. Viruses have a variable host range, but they tend to be rather specific. They tend to be specific to a relatively narrow range of hosts, sometimes an extremely narrow range of hosts. And within the host, they have a range of cells which they will infect. And all of their infectivity is determined by their ability to bind to receptor proteins on host cells. That is the very first stage of viral infection, is binding to the host cell. And they have one or other recognition mechanism on the outside of the virus, which allows the virus to bind to specific proteins, specific substances on the cell membrane of the correct host cell. That's how the virus finds the correct cell. We looked at a couple of different examples. So, for example, very narrow range, HIV only infects human beings and only infects certain cells of the human organism. And those are cells with, in particular, with a protein called CD4. And then there's one other protein which is required, which we will discuss when we talk about HIV disease. That's a very narrow range. There are others. Measles, for example, infects all of the great apes. Rabies virus can infect any mammal, and so on. Varying ranges of hosts, and within the hosts, very specific range for particular cells that they infect. So let's have a look then at some of the structures of viruses. And you'll see that at its very simplest, a virus consists of a protein coat, shown here, this sort of geometric shape here. And this is a protein coat made up of protein units. Each protein unit, is called a capsomere. And the whole protein coat is called the capsid. Here it is in the electron micrograph here. And enclosed in that capsid is the nucleic acid, whatever it is. Viruses have various genomes. There can be DNA and it can be RNA, or it could be, it depends on the virus. But they are always here enclosed in a protective protein coat at the very minimum. So this is a very simple virus. This is actually rather similar in some ways to some of our cold viruses, adenoviruses. So that's this very simplest. One of the things to note is that how these capsomeres fit together. They fit together to form a crystalline shape to the capsid. And we are actually able to crystallize viruses out from solution because of the structure like this. So these are the capsomeres all going together to make up this capsid, the very specific shape containing whatever nucleic acid it is on the inside. So we call that a non-enveloped virus because it is a naked capsid. And by the way, these, in that case, sometimes they may have some of these capsomeres here, depends on the virus, maybe spikes, which are recognition. But in the very simplest viruses, it is the capsomeres which do the recognition of the host cell. So here is the alternative. This is an enveloped virus. And an enveloped viruses are very typical of animal viruses. And this is a little bit difficult to relate that to what we've just seen, but I'll point it out to you. Here, in fact, is the capsid, this blue tube here. is the capsid in this case. And they're again made up of the capsomeres, the protein subunits that link together to form this tube shaped, and in this case, helical tube, which is curled up here inside this envelope around the outside. Now, this envelope here has a special origin, which we'll discuss in a minute. But this is very typical of animal viruses, that the capsid of the virus, here's the helical strand of nucleic acid in the middle here of the virus inside the capsid. But the whole thing is surrounded by this envelope, which is actually derived from one or other of the host membranes. It is modified. It is modified because it has viral proteins embedded in it, but the membrane itself is derived from the host. So here we have the capsid surrounding nucleic acid again, but the capsid itself is now enclosed. And we may have further elaborations on these basic structures, as you'll see in a minute. So here is this is actually a naked virus. It's an unenveloped, but helical. And this is what makes it helical. The capsomeres here fit together, not as we saw them in that kind of geometric shape before, but they are fitting together in a spiral, in a helix. And you can just see, the helical shape here, spiraling round like this. And it actually, very often, the nucleic acid is actually associated with the inside of those capsomeres. So it also is twisted into a helical shape inside. So these are the capsomeres making up this capsid. The capsomeres laid down in a spiral fashion. This is very typical of a virus, which I'm sure you've heard about, and this is Ebola. Ebola is one of the longer of the viruses that we will see. And here it is here. You can't see the capsomeres here, but they would be all spirally laid down to make this a helical virus. And Ebola has this very typical end to it. It's called, some people refer to it as a carpet beater. And a carpet beater was a wooden thing shaped like a tennis racket almost, which you used to use in the old days to beat carpets, to get the dust out of them. And that's what the Ebola virus typically looks like. So here again, that virus that we saw before, the very first one, actually has a particular shape, and we call it icosahedral. And it's again here the capsomeres on the outside, and here on the inside the nucleic acid, whatever kind it is. Here's a typical enveloped virus, and this, by the way, would be very typical of several of our viruses, in particular HIV. This or one of this HIV or one of the herpes viruses. These all have this basic plan. And now you can see in more detail that this envelope on the outside is actually a membrane. It is a membrane derived from the host. It has been highly modified during the viral infection. And one of the things that has happened is that a number of these spikes, these protein or glycoprotein spikes, have been embedded in that membrane, in the envelope. We'll see how the envelope is invested around this capsid on the inside in a minute. This the capsid shape of the capsid varies, by the way, and you'll see that it's slightly different shape in HIV, but the basic plan is exactly this: an envelope surrounding a capsid, which surrounds the nucleic acids on the inside. Now, those actually are relatively simple viruses. It's typical for many of our viruses, but when we get to the viruses that infect bacteria, we can see some very, very complicated structures. And these are called complex viruses for the simple reason what we saw before. We saw the icosahedral, the geometric shape of some sort, and we saw the helical, which we saw in Ebola. We saw in the animal viruses this kind of icosahedral head surrounded by a membrane. But now in some of the bacterial viruses, the bacteriophages, we see a combination of these structures. Not enveloped, but here is the icosahedral head here, and here is a helical protein structure here, forming a sheath below. Now these, we do actually also see some of the complex viruses in animals as well. This is an orthopox virus. Don't worry about it. We'll just remember that it's there. But these complex viruses are extremely interesting, and these are typical of many of the bacteriophages. And you have to realize that when the bacteriophage has to do several things. First of all, it has to recognize the correct bacterium. They actually tend to be very, very specialized often, not just for one species of bacterium, but even for a single strain of bacterium. And as a result, they have very careful ways of trying to recognize their host. Sorry, I can't seem to go back. Here we are. They have to have very careful ways of recognizing their host, and they have to stick to the host. The second thing is that they have to get their nucleic acid through a thick cell wall and through the cell membrane. And they have to do it really quickly and efficiently, because the bacterium actually has mechanisms which recognize that it is being infected and does have ways of fighting off the virus. So here's an electron micrograph. This is showing these complex viruses attached to the outside of a bacterium. Here's the bacterial cell wall here. Here is the virus. You can see the icosahedral head. You can see the shaft, the sheath rather, there. And then these tail fibers here. So this is the most cunning mechanism, because what this is, in fact, is a little injection syringe. So at the head here, this is a DNA virus. It has DNA as its nucleic acid. The DNA is carefully packaged inside the capsid here. Then there's a long tube that runs through here with this helical sheath around it. And under normal circumstances, this is like a spring, and it's expanded. It's cocked, in other words. And when it's cocked against this base plate here, so it extends down like this. Then all these tail fibers here, these tail fibers actually are the proteins that are going to recognize the bacterial host and stick to it. So once they successfully bind to the correct host, these legs, these tail fibers collapse, and they push this base plate down against the cell wall. And at the same time, this spring collapses, and this tube gets forced through the cell wall of the bacterium and through the cell membrane into the cell. And as soon as that happens, all of the DNA is pumped through into the cell, into the bacterial cell. It's a very, very cunning mechanism, and a very, very complex structure. So, and we will see this again because it's typical of the bacteriophages. So have a discussion now about bacteriophage and about how we actually have shown the biology of viruses much through the study of the bacteriophages. The first thing to say about viruses in general, working with them in the laboratory is rather difficult because they have to be grown, they have to be cultured in living cells. Unlike bacteria, which can be cultured on inanimate media, the viruses have to be cultured in a living system because they depend upon a living cell for all of their reproduction, because they are entirely parasitic. So what we usually, for bacteriophages, what we usually do is we will grow them on a lawn of bacteria in a culture structure. You can do it in broth, but they are most easily visualized if you grow a lawn of bacteria on an agar plate. And when you do so, you can get a solution of bacteria, of viruses, diluted heavily, and spread that together with your bacteria. So what you're seeing here, this opaque background here, is the lawn of bacteria. It's bacteria that have grown so that you can't see single colonies. They're all just in a thick layer, abutting on one another. But wherever there's a virus present, it will infect bacteria. It will reproduce inside a bacterium. That bacterium will release the viruses in one way or another. And those viruses then spread around and they begin to infect the bacteria nearby them. And pretty soon they will have cleared little patches here, which are visible as clearings in this background of bacteria. And these we refer to as plaques. I say plaque, other people say plaque. There's no, either one is correct. And this is very useful because this means that we can actually, if we know the dilution factor that we had for our viruses, then we can work out the concentration of virus in our original sample. The same way that you did in a way when we counted bacteria using a spread plate. So here we're counting the number of viruses by seeing how many of these little spots or plaques they form in a bacterial lawn. So, growing bacteriophage is relatively easy. Growing animal viruses is a lot more difficult. And this was an impediment to the study of viruses for many, many, many years, until systems were developed whereby we could actually grow viruses reliably. One of the ways, of course, that you can do it is to infect living animals. And so this is done often using mice or rats or rabbits. Those are the three animals, really laboratory animals of choice for viral studies. But it is also possible to grow viruses in cell culture, providing that the cell culture you're using is within the viral host range. So that is another way to do it. And once these methods of cell culture had been developed from 1910 on, then viruses began to be cultured in cell culture as well, and they still are very much so today. That's a common way of doing research on viruses. But one of the most useful techniques is to grow viruses in growing hens or ducks' eggs. For some reason or another, duck egg and hen's eggs are very tolerant of many different viruses up to a certain stage. It's just a freak of nature, really. Many viruses can be grown, at least for a time, in growing poultry eggs. And this is probably the commonest way of growing virus. And in many cases, a vaccine is developed through the culture of viruses in hens and duck egg. So this also gives us a chance of monitoring, if you're growing virus in in an embryonated egg. Gives us a way also of monitoring changes in cell development and everything in the egg, because there are ways of visualizing the interior of the egg. All that you do is you very carefully crack off, without injuring the membranes underneath, you crack off the shell, you lay a Vaseline on top of it, and it goes clear. And you can actually look at the embryo using a dissecting microscope or whatever, and you can monitor changes. You can inject virus, see what happens to the embryo, see as you can watch as the virus spreads, etc. But it is also a way of increasing viral population if you want to develop a culture of viruses to a certain concentration. Growing them in poultry eggs is one of the classic ways in which this is done. So there is considerable debate about how to identify particular viruses. It is not always an easy matter. The first thing is we do, of course, if we are able to culture the virus. It's not always the case, but if we can culture the virus, the first thing is that we can examine the morphology. This wasn't always able to be done because, of course, viruses cannot be seen with the ordinary light microscope. But using electron microscope techniques, we can define morphology of viruses. But there is, in fact, a somewhat limited range of morphologies. Viruses tend to fall into very broad categories based on morphology. So that's not always that useful to, it's useful, but not always definitive to know what shape its morphology that a virus has. Instead, we do depend on the fact that viruses produce definite, repeatable symptoms in host animals or in cell culture or in embryonated eggs. The results of infection are reproducible. We see the same kinds of symptoms from specific viruses. So that is one thing which can be used to identify a virus, actually the symptoms that the virus produces. This is not always that definitive. And I'll give you a simple example. The influenza virus in us produces symptoms, which are some symptoms which are almost identical to the symptoms produced by nearly 200 common cold viruses. That is why people are always often get influenza and a cold and colds mixed up, because we can't tell that much from the symptoms alone, from some of the symptoms alone. You have to look at the whole scope of the disease in order to define by the illness that it produces. In culture, for example, in cell culture, we can recognize cytopathic effects, specific changes that take place in the cells that are infected. The third way, really important way, is for viruses which have been defined and which have been used and characterized in the laboratory. We can use serological tests. That is, we can, for example, we have a virus, a known virus. We can inject that virus into an animal, a rabbit, whatever, have that animal produce antibody to that virus. Then afterwards use that antibody. Antibody is extremely specific. We can use that antibody to test unknown viruses. If it reacts against those unknown viruses, then our unknown is the same as the virus which produced the antibody in our experimental animal. These are serological tests, and they are standard tests you may have heard of. For example, ELISA tests are one big group of clinical laboratory tests which are done for HIV, also for HIV herpes, all sorts of things. We do ELISA tests, and those are serological tests using antibody to from a known source to identify an unknown virus. The last way is one which is relatively new, and it's absolutely now of critical importance. And that is being able to measure the abundance of specific nucleic acid, of specific genomes present in a host. This is what you've heard of for COVID, for example. COVID is actually a way of measuring the presence and abundance of the COVID genome in the human body. We do the same thing with HIV and with many other viral diseases. We actually use techniques such as the polymerase chain reaction, PCR, to develop a picture of how much nucleic acid is in circulation in the host, which is tied to a particular virus. So that is now probably one of the most refined and important techniques used for identifying viruses. Okay, so I will tell you that this is identification of viruses is a mare's nest, and is the source of very great controversy in scientific circles, as we try desperately to provide a classification system which allows easy identification of the placement of viruses within certain categories. One of the reasons for that is that viruses are not living entities. So their evolution and everything is very complex, and it does not work according to the sort of genetic rules that other organisms obey. There is an international committee on the taxonomy of viruses, which works to recognize and classify particular viruses and give them a name which defines them. So, for instance, COVID is in actual fact SARS, severe acute respiratory syndrome, SARS-CoV-2. And it's a COVID virus, CoV, COVID virus, and it is the second one to have been recognized. SARS-CoV-1 was what is commonly known as SARS. SARS-CoV-2 is our current pandemic of COVID. That name is decided on by this committee, and it serves to define that particular virus. And they will define it in terms of various categories that we have already discussed. So we're going to now just go through some of the categories. I'm not going to do it in enormous detail, but I'm going to mention some of the common viruses that you will have heard about, and I'll tell you some of their characteristics. So the very first thing to know is that viruses can have one of two kinds of genome. Viruses can either have DNA or they can have an RNA genome. That's one of the reasons they don't fit in with the rest of living things, because we define living things on our planet as depending upon DNA as their nucleic acid. That is not the case with viruses. Can be DNA, depends on the virus, can be RNA as well. There are many RNA viruses, as you will see. The other thing to tell you is that if there is a variability possible in the genome, viruses have it, and you'll see what I mean. We are used to the idea already that, for example, DNA is a double-stranded nucleic acid, where RNA is a single-stranded nucleic acid. Well, viruses completely ignore that rule. There are many DNA viruses which have double-stranded DNA. That is true. But there are also viruses which have single-stranded DNA. This is absolutely extraordinary, but it is true. It is just a single strand of DNA. It doesn't have matching bases on the other side. So we'll just mention that. You'll see in a minute. Of the DNA viruses, there are an enormous number which are very important to vertebrates and to human beings in particular, as we'll talk about now. And most of all of those that are important to human beings are actually double-stranded DNA. Let me just mention the single-stranded, just to get it out of the way. All I need you to remember is that single-stranded DNA viruses actually do occur. Those of you who have dogs will have heard about one of them, and that is this is parvovirus. The parvoviridae are particularly important in certain groups of animals, and dogs are one of them, where it causes a very serious disease in dogs. So just remember, parvoviridae are single-stranded DNA. It's very strange and extraordinary. And these viruses here are non-enveloped. It is just the capsid and then the single-strand DNA inside. All of the other DNA viruses we're going to talk about are double-stranded, the normal structure of DNA. But we recognize that some of these double-stranded viruses are unenveloped. It's just the naked capsid. Others are enveloped and are wrapped around with a membrane derived from somewhere in the host, as we'll learn in a while. So let's have a look. We've got double-stranded, and now we're going to have a look at the non-enveloped, those with just the capsid on the outside. There are some very important human viruses that fall into this group. These are the adenoviridae. The adenovirus, they cause a wide variety of illnesses. It depends on the adenovirus, but the commonest one that we encounter is a common cold virus, the adenovirus, which is a common cold virus. And that's the first thing to note of importance. There are other adenoviruses which do more serious things. There are some which cause viral meningitis, which is the viral meningitis is an infection of the membranes of the brain. It causes inflammation of the meninges. Viral meningitis vary in severity, but in general, viral meningitis is not a particularly serious illness. It is unlike bacterial meningitis, which is extremely, extremely serious. So those are the adenoviruses. The only other thing I'll mention about the adenoviruses for you is that the adenoviruses can act and are used as vectors for vaccines and for other things as well. We can use adenoviruses as little traveling spaceships to carry useful information to cells. I'll talk about that much later on. Just put that into your brain somewhere. We can actually manipulate adenoviruses to do useful things for us in medicine. The other group of non-enveloped DNA viruses that we need to hear about are things like the papillomaviruses, papillomaviridae. Now, papillomavirus is extremely common, but there are many different strains with different clinical effects. And I'm not going to go through all the details of the illnesses at the moment. I will just tell you that papillomaviridae include the common warts, which are more common in children, do occur throughout life, but are more common in children, just the benign warts on skin. But papillomavirus is also responsible for genital warts. It is a strain of the papillomavirus which causes genital warts in both male and female. And the genital warts papillomavirus has the potential, it's not inevitable that it will do so, but it has the potential to cause cancers, both in male and in female. Much more commonly in female, it causes cervical cancer. And in males, it can cause penile cancer, but that is relatively rare compared to cervical cancer. And you'll probably know that there is now an excellent vaccine, which is available to both men and both male and female, should be administered before puberty, and then shortly after puberty, again, a booster shortly after puberty starts. But it is an excellent vaccine, which has, where it's been tracked, has produced prodigious decreases in the rates of cervical cancer. So those are the papillomaviridae, non-enveloped, double-stranded. There are far more double-stranded DNA viruses which are enveloped, which are important to us. The first are the pox viruses. Now, the pox viruses occur widely throughout mammals. There are many different kinds of pox virus, but they tend to be very, very specific for a particular host. So, for example, smallpox, which was present in the human population right up till 1976, is specific for human beings. It is transmitted only from human beings to human beings. And the disease of smallpox was eradicated from the planet, essentially, by 1976 through a rigorous program of public health and of vaccination. And I'm willing to bet that none of you probably none of you have been vaccinated against smallpox, because it's not necessary anymore. The disease does not occur on the planet because of the successful vaccination campaigns. So those are the pox viridae that had been extremely significant in human populations and had been the cause of many great plagues of smallpox. Still very common, however, are the herpes viruses. And the herpes viruses vary from those which cause disease, which is just mild nuisance level disease, such as cold sores, to causing very serious childhood illnesses, such as chickenpox, and also very serious adult diseases, both chickenpox, if it's acquired as an adult, or shingles, which occurs in people who had chickenpox and have a recurrence of the virus. Then various other diseases, you might have heard of mononucleosis, for example. This is also a herpes virus. There are many different diseases caused by herpes virus. It's a very, very large group. Depends on the herpes virus that you're talking about as to the disease it causes. They are enveloped, double-stranded DNA viruses. They have one other characteristic. Most of the herpes viruses are able to remain, after an initial infection, are able to remain latent, usually do not reappear at all throughout the lifetime, but can do so. Chickenpox is a good example. Chickenpox, when you have it as a child, it retreats and remains completely silent actually in your nervous system, in peripheral nerves, and then can reappear as this disease, shingles. That is characteristic of the herpes. Many of the herpes viruses, they have a tendency to be able to remain latent. They are enveloped, double-stranded DNA viruses. Now, I'm just going to mention one other one, and that is this one here. These are the hepadnaviruses. Very, very strange viruses indeed, very strange reproduction. They are kind of a cross between DNA and RNA viruses. They are formerly DNA viruses, but they replicate in the cell. They replicate through an RNA intermediate. So those are hepadnaviridae. Just remember they are that HEPA part, the hepadna part, tells you they're mostly associated with liver disease. But that's all I think I'm ever going to say about the hepadnaviridae. We'll spend more time talking about these than we will about that. So those, for the moment, those are our DNA viruses. There are many more. I'll tell you, this is not all of them. This is just a brief overview. So here are these now are viruses which have an RNA genome. And there's something that I need to explain about the RNA genome. I told you if there's a variation on these things that is at all possible, then the viruses have it. And here's a good example that we saw in our DNA viruses. Most DNA viruses are double-stranded because that's the normal DNA structure. Some of them are single-stranded. Well, look here. Here we have RNA viruses. We learned that RNA is a single-stranded molecule. Fundamentally, it is. It's a single-stranded molecule. Most RNA viruses have a single-stranded RNA genome, but there are viruses which have a double strand. And I simply want you to remember there are viruses which have an RNA genome, which when you look at it, superficially, it looks like DNA. It is double-stranded and it is helical. But if you were to look at the sequence, you would realize that it does not have thymine in it. It has uracil. So it is actually double-stranded RNA. And that's all I just want you to remember, that they exist. The most important group are the Bio3300 which viruses which cause a gastroenteritis. So we're not going to spend much time talking about them. We'll talk about single-stranded RNA instead, because it's such a huge group. And for human beings, it really is probably the most significant of the viral groups. So this is just a normal single-stranded RNA. I hate to introduce another complication for you, but there are two ways in which you can have an RNA genome, a single-stranded RNA genome. The first is if that RNA strand is just like messenger RNA. So I take that this single strand, put it into a cell, and when the cell sees it, it sees it as messenger RNA, and it will immediately begin to read off and translate and produce proteins. Its ribosomes will grab onto it and will start to produce proteins from it. So it's just like a messenger RNA strand. That is called positive strand, that positive strand RNA. But you can have the matching strand. If you imagine, I took an messenger RNA strand, and I made a duplicate strand with matching bases. Okay, so if my messenger RNA strand read AUG, then my matching strand on the other was UAC, right? So that is called negative strand. And I'll tell you when we get there an implication of this. But this positive strand, if I introduce this viral strand into a cell, then the cell will recognize it as if it was messenger RNA and begin treating it as messenger RNA immediately. Ribosomes will latch onto it and just start producing protein. So some of these are very, very well known. You'll recognize. Again, we have the same distinction. They can be enveloped or they can be unenveloped. So here we have single-stranded positive strand RNA. Now let's just talk about those first, which are unenveloped. And the only group I really want you to know about are these, the picornaviridae. Picornavirus tends to be a rather small virus. Many of them cause the common cold. There are several which will cause symptoms of the common cold. One very, very important one in human beings is polio. Poliomyelitis, the paralytic disease, usually of childhood, not exclusively, but usually of childhood, and it can affect all primates. And wild primates, chimps, gorillas, are infected through human beings. It does not exist wild in their population, so they acquire it if they are in contact with human beings. They can acquire it if they're in contact with human beings. Poliomyelitis, again, responds very, very well to a vaccine. There are excellent vaccines available. And this is one of the diseases which we are on the point of eliminating from the human population. It should happen certainly within your lifetime, well within your lifetimes. So this is picornaviridae. The only other important picornavirus is this one, which is hepatitis A. And hepatitis A is quite a nasty, but very seldom dangerous illness. It's an inflammation of the liver that produces temporary incapacity for a period of weeks usually, but usually people recover completely from it. That is caused by a picornavirus. So those are typical non-enveloped. So here we come now to the enveloped positive strand, single-stranded RNA viruses. And here you'll hear some which you have certainly heard of before. The first group are the togaviridae. And togavirus, the one that you may have heard of is rubella. And rubella, because it seldom occurs now in most developed countries, because there is a very, very good vaccine, measles, mumps, rubella vaccine. Rubella, the reason that first of all, I'll tell you, rubella is a really mild illness in children, usually. There are always exceptions, of course, but in general, it's a very mild illness in children. So why bother to get a vaccine? Well, the reason is that if, first of all, immunity from childhood does not last particularly well. And if you've only acquired it from the illness. And in pregnant women, if they get rubella, they may not even know that they've had it, but at certain stages of a pregnancy, it can induce catastrophic developmental error. So children are born with massive physical defects, for example, as a result of rubella infection of the mother. This is the reason that we give rubella in childhood. The vaccine produces a very, very long-lasting immunity. So the incidence of rubella has fallen tremendously in developing countries. The next group are called the flaviviridae, and these are certainly, some of these you will have heard of because they are viruses which are of current interest, especially with global climate change. The ranges of these viruses is expanding because many of them are viruses which are vectored, especially by mosquitoes. Many of the flaviviridae have a dual host range, and that is between mosquitoes and other animals, especially humans. And here are some you will certainly have heard of if you've paid attention to the news in the last couple of years. Yellow fever is one of them. Dengue fever is another, and the other is West Nile fever. And West Nile fever for us is of particular interest because it has been recorded very close to New York, as the range of its mosquito hosts has expanded. It is present in southern states and its range has expanded, and it may, in fact, it is predicted that it may reach as high as Connecticut eventually. The other important flavivirus is the one that causes hepatitis C. Now, hepatitis C is a disease, not very contagious, and it is generally acquired through intimate contact, either sexual contact or especially through transfer of blood products. If people are sharing needles, for example, hepatitis C is easily spread. Hepatitis C is one of these diseases which sets up a chronic infection and can lead to liver collapse, eventual liver collapse, and liver cancer as well. It's a very nasty virus, but interestingly enough, it is one of the few viruses now for which we actually have a drug cure, which is available. Next group. Well, should we just skip it over and pretend that it doesn't exist? And unfortunately, we can't. This group of single-stranded, positive strand enveloped viruses are the coronaviruses. And the coronavirus, of course, is a group that our current SARS-CoV-2 belongs to. That is the virus which is currently causing COVID, the COVID pandemic. This is a coronavirus. There are a number of these coronaviruses which are known to cause illness in human beings. The two SARS-CoVids are SARS-CoV-1, which causes the disease of SARS, a very, very serious illness with a very high mortality rate. And then SARS-CoV-2, which is much less infectious than SARS-CoV-1 and is much better tolerated in many people, although, of course, as you know, it can also cause serious illness and death in about 2% of the population. People that have it acquired. The other diseases associated with this, there are several others. There is one called Middle East Respiratory Syndrome, for example. And there are a number of the coronaviruses which just cause the common cold, cause symptoms of the common cold as well. There are 4 of them, I think, that are recognized. And so this is very interesting because it appears that these coronaviruses enter our population with regularity and then end up simply being tolerated as relatively mild illnesses over evolutionary time, thousands of years. We end up adapting to one another. That's what it seems. So when we look at things like SARS-CoV-1 and SARS-CoV-2, what we may be seeing is the entrance of new coronaviruses that will eventually, the human population will adapt to, hopefully. The next group are extremely interesting. This is, again, these are single-stranded, positive strand enveloped viruses. But this is a group which has a very special reproductive strategy. All of these here, this is positive strand messenger RNA. When it enters the host cell, essentially it just looks like messenger RNA. And through one mechanism or another, what the host cell does is it uses this messenger RNA. It first of all reads off many, many copies of the messenger RNA. And it also reads the messenger RNA to derive all of the viral proteins. And then it assembles all the proteins, puts the nucleic acid together, and boom, you're done. This group here, the retroviruses, have another little twist to their reproductive tail. And that is that when the single RNA strand is injected into the host cell in the retrovirus, they actually read that messenger RNA strand and produce a DNA strand from the RNA strand. We'll talk about how that is done much later on. For the moment, just realize this is why these are called the retroviruses. The retrovirity, the backwards viruses, in other words, because what they're doing is they are reversing the central dogma. What does the central dogma say? DNA is transcribed to messenger RNA, right, which is translated. DNA is transcribed to messenger RNA. Well, in this group of viruses, what they're doing is they're forcing cells to take the messenger RNA and reverse transcribe it. They take messenger RNA and they reverse transcribe it to DNA. They then take that DNA and they shove it into the host genome and turn it into an HIV factory eventually. But their first reproductive step is to reverse transcribe their single-stranded positive RNA genome. So they are called the retroviruses. Okay, so let's proceed. We're still on single-stranded RNA, but now we're talking about negative strands. And all that has to happen now is the genome of negative strand viruses is put into the host cell. And one way or another, there are various strategies for doing it. A positive strand, a messenger RNA copy, is made from the negative strand. And then reproduction proceeds. But there has to be this additional step for the negative strand. The negative strand, a copy is made, which looks like messenger RNA. And once that messenger RNA strand is present, then the reproduction of the virus will proceed. Again, in this group, we have enveloped and unenveloped, but we're not going to talk about any unenveloped. We're only going to talk about the enveloped viruses. They're the only ones of importance. Some of them you will definitely recognize. The first group here are called the filoviridae. And let me just show you. Here's why they're called philo, means a thread. There's why they're called the filoviridae, because they are thread-like. And this is Ebola and various related viruses. The filoviridae in general produce hemorrhagic fevers and vicious, very, very dangerous hemorrhagic diseases indeed. This is typical of Ebola, that people effectively end up bleeding out through virtually no apparent cause. They bleed from the skin, from the eyes, from the ears, the gut, and they just literally leak blood as a result of these infections. They also, the filoviridae, have the characteristics, they are very, very, very contagious. So this is interesting in epidemiological terms, because you'd think with such a fierce virus producing such fierce disease would surely have a great propensity for funding a pandemics or epidemic pandemics, right? It sounds like it should just run rampant through the human population. Well, it doesn't. These tend to be very localized. And one of the reasons is that they cause such serious illness. Two things happen. People get ill very, very quickly and they stop moving. The virus is definitely spread by people migrating from one place to another. That is true. But in general, people, once they fall ill, they stop. And the contagion spreads very quickly. People become ill very quickly. As a result, what you'll find is it'll be a village or a group of villages where the disease, and it is so distinctive that people tend to avoid it. As soon as there's word that somebody has Ebola in there, they tend to desert the area. They just stay right away. And for this reason, these tend to be occur in very spectacular, limited epidemics, not spreading into pandemic status, largely because of the virulence of the virus itself. Let me see if there's I don't want to talk about Banya virus. That's okay. The rhabdoviridity we have already heard about. Again, a single-stranded, negative strand and enveloped. Rhabdoviridity, these are the rabies viruses. And the rabies virus, rabies can occur in any mammal, and it's a neurological disease. In some mammals, it's much better tolerated. But in human beings, essentially once symptoms begin occurring, it is almost 100% fatal once symptoms start developing. Fortunately, it progresses initially very, very slowly. And so there is a lot of time to effect some treatment if you know that somebody's been exposed. The next group are the orthomyxoviridae, probably one of the most important human viruses of all, still causing millions of deaths per year, and at times has produced great pandemics, which have resulted in catastrophic mortalities of the human population. And the orthomyxoviridae, these are the influenza viruses. Notice they are completely separate from anything which would cause a common cold. They are in their own group with their own. The orthomyxoviridae have very, very particular characteristics. And I'll just mention for the moment, there's a negative strand RNA virus, but that negative strand of RNA is cut up into 8 pieces, into 8 separate pieces, each with its own little capsid around it. And it's a strange genetic arrangement, but the strange genetic arrangement allows the orthomyxoviridae to hybridize with one another, different kinds to hybridize with one another. And there are 3 principal hosts, and they are poultry, ducks, chickens, etc., but especially duck, pigs, and human beings. Those are the 3 principal hosts for the orthomyxoviridae. And those 3, they basically have their own groups of flu viruses, but they can mix and match between the 3 with catastrophic results for populations. So that we'll talk about when we get there. The next group you will also have heard about, and these are the paramyxoviridae. And these, because they are very common childhood diseases, and they can have extremely serious consequences. And you will have heard of mumps and of measles. Of the 2, measles is probably the most virulent and has the most likelihood of producing serious sequelae, even in a childhood infection. And so these 3, mumps, measles, and rubella, are commonly vaccinated against together in early childhood. And of the 3, measles is by far the most virulent and the most dangerous of all of those diseases. Arenoviridae, no, we don't need to worry about. Okay, so for those that I've mentioned, just go through this again and maybe make a list. Okay, make boxes for each of these categories, and then list the viruses and some brief characteristics. We will hear about some of them again, not all of them, but we'll hear about several of them again when we talk about viral disease itself. Okay, so let's talk a little bit just about how viruses actually do multiply and how they reproduce inside their host cells. Well, the very first thing is that a virus has to be able to invade its host cell. And once it's invaded the host cell, then you can set about taking over the host machinery, etc., and effectively turning the host cell into a viral factory, which just produces more viral particles, which have to escape somehow and then go on to infect more cells. When we look at the viral growth curve, we see that it actually looks somewhat different, say, to the growth curve of the bacteria that we saw before. It's really a one-step growth curve, where before we had that complex curve, the lag period, then exponential growth, then a stationary period, and then a period of decline and death. Here, instead, what we actually see when, if we are monitoring a virus that's present in a person, let's say, who is suffering from an illness, and we look at the number of viral particles. The virus itself, the viral particle, is called a virion. It's not a cell. It has its own name, the virion, and that is the unique independent viral particle that is going to cause infection. So if we count the number of virions that are present, initially we may actually see quite a lot, because there has to be an initial dose. It varies from virus to virus. Measles, for example, is very, very low. COVID is reasonably high. And then other viruses, it actually has to be really high for us to be able to establish an infection. There is an initial period where you might pick up quite a lot because the person's been recently infected, but the number of virions actually disappears because it drops. Because what happens is, first of all, the body just eliminates a whole lot of the viruses. They get stuck in your mucus and get sneezed out or whatever. And the others that are present and able to establish infection actually disappear because they've established the infection. They're inside cells. We don't see them anymore. We're counting a virus in serum or whatever. We don't see it. This is called the eclipse period. But while this is happening, virus is actually being reproduced inside the cells, preparing for escape from the host cell. Initially, you've got a limited number of host cells that are infected, but they release a large amount of virus. That virus goes out and infects more host cells. This is in the same host. It infects more cells. They, in turn, release more and more and more and more. The number of virions present begins to increase and then increase and increase and increase. Then there's a period of acute infection. That is the period when the virus is being actively produced by cells, actively released, and is also the period probably when the immune system is beginning to recognize what is happening. And once the immune system responds adequately to the presence of virus circulating in the body begins to drop, and eventually it falls to zero. So if we follow this, it's somewhat different to that growth curve that we saw before. But we also need to realize that what it shows us is that during this period of acute infection, when cells are releasing the most virus, this is also the period when the most damage is being done to cells, but it's also the period when your immune system is most likely reacting most strongly to the presence of virus. This is the period not only of acute infection, but it's probably also the period of acute illness. And it's partly because of viral effects, direct viral effects, partly because of the immune system effects. So a lot of what we know about virus reproduction, we have learned from studying bacteriophage, those viruses which infect bacteria. And there are some parallels to be drawn to other systems as well, as we'll see. But let's just concentrate for a while on bacteriophage and how they operate inside bacterial cells. I'll tell you first of all, we recognize two different kinds of interaction. The one is a catastrophic, acute kind of reaction by bacterial cells. And this is called the lytic cycle. In the lytic cycle, virus infects the bacterial cell, goes through a round of reproduction inside the cell, the equivalent of the eclipse period inside the cell, and then actually causes the cell to burst, produces masses of virus particle inside the bacterial cell, then bursts, and kills, obviously, the bacterial cell, and releases the viral particles. That's a lytic cycle. There is another kind of cycle called the lysogenic cycle. And in the lysogenic cycle, something a little bit different happens, because the genetic information of the virus becomes incorporated into the host DNA. So it forms part of the host DNA. And there may be a lengthy period when nothing else happens, when the viral DNA, viral genes are not read off. They just get reproduced. Every time the cell reproduces, it reproduces the viral DNA as if it was part of its own genome. But then through some other event, often an environmental cue, the virus kind of reactivates. The virus converts. We call it phage conversion. And it now enters a specialized cycle, which is going to end up causing lysis. So here is a typical bacteriophage. You'll recognize the structure because this is a complex virus. So here's the capsid there. There's the sheath. Here are the tail fibers. Here is the tube that I told you about, which we didn't see before. This tube has poked its way through the cell wall and the cell membrane into the bacterial cytoplasm. And the DNA here is being injected. This is a DNA virus. This is a famous bacteriophage called T-even. We'll hear about one or two others kinds in a while, but this is the T-even bacteriophage. And T-even causes a lytic reaction. So the first thing is, we've explained already, the virus attaches to the cell wall by the tail fibers, recognizes the correct host cell type through those tail fibers, and attaches. Those tail fibers collapse. The phage pokes a hole through the cell wall, through the cell membrane. By the way, this tube doesn't only go through mechanically. The virus actually has its own enzymes which can digest away the cell wall to facilitate it. It injects the DNA into the bacterial cytoplasm, and immediately it co-opts the genetic machinery of the host, and the host genome begins now. The host cell machinery now begins to produce, first of all, replicas of the viral DNA and all the requisite proteins to make up the virus. Those proteins are assembled inside the cytoplasm, and the phage DNA, the viral DNA, is packaged inside the capsid. And once that happens, then the bacterial cell is induced to burst. And it bursts open and releases all the phage particles. So that is relatively easy to understand. Here it is here. Injects the DNA. The DNA actually, in this case, in these phages, they usually break up the host genome. but the kind of the transcription and translation machinery carries on and operates on the viral DNA. It produces all the proteins and lots of copies of the viral DNA. That all gets packaged up together. And once that happens, they actually cause the cell wall to burst. They release viral enzymes, which cause the cell to burst and simply release the particles. Those go on, they go on and infect other bacterial cells. We saw that. We saw that here. What happened here? Exactly that. We had the lawn of healthy bacteria. We infected them with T even. One cell maybe in the middle got infected with the virus, lysed, released those particles, and they spread out around. And then those cells bred virus, burst, died, released, and it begins to move out until it forms that visible plaque that we saw there. Okay, so that's the one kind, that's T even. Now here's another virus, and this is called the lambda. Lambda is one of the most famous of all of the viruses, of all of the bacterial viruses, and simply because this is where this was elucidated, this life cycle was elucidated, but also because bacteriophage lambda is very useful in genetic engineering and is widely used in genetic engineering. So this term lysogeny, it implies that the phage is actually able to remain latent. It can lie, but it's for much of the bacterial cell's life cycle, the virus just remains completely silent. It remains latent. And the reason for that is that instead of immediately beginning to produce viral particles, the DNA is actually incorporated into the host DNA. It becomes part of the host DNA, and there's no other sign that viral infection has taken place. That insert of DNA into the host genome is called the prophage. And it's prophage because it's preparatory to the formation of the viral particles, the phages themselves. Whenever the DNA of the host is replicated, the viral DNA is also replicated. But eventually, through in some environmental cue or other, there we are going to see that this shift from being this latent, silent, and essentially harmless incorporation to being a lytic phase where the virus actually gets reproduced. So here's the start of it. And here's the lysogenic phase. The DNA, the phage DNA is inserted, and it actually becomes incorporated. It's a very complex process, by the way, this incorporation, so we won't discuss it. But it becomes incorporated into the host DNA as prophage. Every time the cell reproduces, every time the bacterial cell reproduces, the prophage is reproduced as well. So it can spread very, very widely within a host population. But then on some environmental cue, this piece of DNA, which forms the prophage, actually excises from the genome. It comes out to look almost like a plasmid there. And what happens now is that the host DNA is inactivated, and instead all of these viral genes are now begin to get read off, produce viral particles, and now the cell enters a lytic phase. This is exactly like the lytic phase that we saw before. And the cell produces masses of viral particles and then bursts. So the difference is, this is the lytic phase, this is the lysogenic phase. Sorry, I'll do the right way. The lysogenic phase here, This is the lytic phase here, and they can alternate. This can go on for a however, lysogenic, can go on for a very, very long time indeed. So it can spread widely within a population. And then on an environmental cue, the switch over to a lytic phase. All right, so let's leave it there for today.